So let's talk about different types of point mutations that we see in our body. You know, point mutation means the mutation in DNA and actually in one particular nucleotide. The change in one particular nucleotide will change the protein expression, because remember what we have DNA as a central dogma. From that DNA we produce mRNA. From that mRNA we produce proteins, right? So what happens here, if any nucleotide change occurs in the DNA level, it will create some change in the mRNA level. So the change in mRNA level ultimately reflect in the protein level, because that mRNA will be translated into proteins, right? So point mutation means the mutation in a single nucleotide. Mutation in a single nucleotide in the DNA, and that will change. That can be in the mutation in the DNA level, that can be mutation in the mRNA level, and finally ultimately reflect on the proteins. Because you know what we know about codons. Codons are 3 nucleotides long stretch. For example, CAT, this is one particular codon which is responsible for producing a particular amino acid, for adding a particular amino acid. In this case the amino acid is histidine. So if there is a codon called CAT in the DNA level, it will incorporate histidine, right? Or if it's an mRNA level, CAT, it will encode histidine. So that histidine is now added. Similarly, for different codes, different amino acids will be added, and ultimately those amino acid will create the primary structure of polyamino acid or polypeptide sequence. Say histidine, proline, serine, arginine, they are linked with each other, they produces a kind of primary structure. Now that structure will be folded to make the secondary structure and tertiary structure of the protein. That is the actual functional unit of the protein. Remember, none of this primary structure of proteins are actual active or functional, because for a protein to be functional it should be folded, and protein folding is still a big mystery. How it's done and all these things, theories are there, but it's not that much clear. But the protein folding is required to have that protein ability to function properly, right? Now what happens here, during any kind of nucleotide change in this, for example, instead of T there is a G placed, that will change this codon. Or for example, instead of this A there is a C placed, that will completely change the amino acid which will be entered inside. So if we change the amino acids, remember there are 20 amino acids, they have different properties. Some of them are hydrophilic, some of them are hydrophobic, some of them have a bulkier group like proline, some of them are very tiny like glycine. So those are different types of amino acids and required to produce different types of proteins. For example, for producing membrane protein, we need to have more of a hydrophobic amino acid stretch to produce that hydrophobic core which will be embedded into the membrane. So during that production, if by any means of mutation we add more and more hydrophilic molecules there, that protein will no longer function as membrane protein. So these are the examples of why this mutation causes such a big difference, right? So if you look at here, for the missense mutation, so let's talk about the missense mutation. In the missense mutation, for example, we take the example of simple element like CAT. Let's say CAT, 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 CAT. For simplicity's sake, we just have a very basic structure here, CAT, CAT all along. Now we know the CAT is very easy to memorize too, because if you all time need some example to draw and write, I believe you draw this. CAT means cat, and cat will produce histidine amino acid instead. Remember that. So cat, 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 this is a stretch of nucleotide sequence in the DNA, for example. Now what happens here, for example, during this stretch somehow they change one of these bases. For example, this particular base got changed. Instead of A, there is a C incorporated. Rest of the things remains the same. CAT, 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 CAT. So remember, this is the DNA normally. After the mutation, the cause of mutation is always the same in different cases. For example, it can be misincorporation of the nucleotide during the DNA replication. It can be misincorporation of nucleotide during the transcription, that is making mRNA from DNA, right? So these things always present. Or during the different kind of physical or biophysical and chemical agents, like UV radiation, there will be production of thymine dimers, like that, right? So these things are always related. For example, during this process, this A, instead of A, during the DNA replication process, the polymerase mistakenly put C instead, and the DNA repair mechanism will not recognize that, they pass this. So ultimately that mutation remains there as a C instead of A. So what it will do? What it does actually, this means histidine, so histidine, the previously we have all of them were histidines, right? All of them were histidines, but now these are histidines, we all know. But during this mutation, these 3-stage nucleotides are termed as the reading frames, right? So rest of the reading frames remains the same, but the reading frame where we inserted C instead of A will change the amino acid to be incorporated, and they now incorporate proline instead of histidine. And proline is an amino acid with bulgier ring that may cause certain problems. So ultimately, instead of histidine, 5 histidine stretch of amino acid, we get 2 histidine and proline and then 2 histidine. So this, for example, this previous protein will fold something like this after this process of translation, but this protein, as a result of this proline, steric hindrance is caused due to the bulgier loop, and protein folding is not possible. So ultimately the misfolded protein will be guide to the protein degradation. So we will have a loss of activity of the protein, or it might produce a malfunctioning protein which does not function what it should do inside the cell. So this thing can occur, and that is called the missense mutation. Remember, missense mutation means missense. Sense means the sense of the codon. Miss means they miss the sensing of the codon. That means the codon should be histidine, but they miss the sense and produces proline. That is called the missense mutation. Okay, and that missense mutation can produce defective proteins which may not function properly, or that can produce proteins which are not folded, will be degraded inside the cell.